All right, so let's look at proteins. Okay, so proteins are part of what we call the biomolecules. Okay, or what we call a biomolecule is simply um, these are molecules that are found in living organisms. Okay, so it's part of what we call the biomolecules. We have proteins, carbohydrates, and then you also have nucleic acids. Okay, and also lipids. Okay, so proteins are part of the biomolecules, and so they are the major components of any living organism. Okay, so when we say any living organism here, we're talking about any organism that's living. Okay, they are the major components. Okay, all living organisms are made up of proteins. Okay, and then just to give a brief definition, they are large molecules. Okay, made up of small units. Okay, so a large molecule, what we are saying is that um, they are very large. Okay, they are, we categorize them being large because we have smaller units that make up these proteins, and the molecular weight is ranging from 5,000 to many millions daltons depending on the type of protein. So proteins are called polymers. A polymer is simply a large molecule made up of small molecules called monomers. So it's a long chain of nitrogen containing monomers, and these monomers are amino acids. Okay, and they are linked together by peptide bonds. So you'll find that amino acid linked to another amino acid, linked to another amino acid, linked to another amino. So it becomes a very long chain. Okay, when this becomes a very long chain, it's called a polymer, and that's what proteins are. So the link is called a peptide bond. Okay, so proteins often can bond with other complex molecules. Okay, so you'll find that you have proteins bonding with the lipids. Okay, and these will be called lipoproteins. Proteins bonding with carbohydrates, these will be called what? An example is glycoproteins. You have proteins that can link together with nucleic acids, these are called histones. Okay, you also have those that can bond with heme. Okay, for example hemoglobin, it can bond with a heme group. Okay, we move further. Okay, so proteins are most important constituents of cell membrane and cytoplasm. Okay, you find them in almost all the cells. Okay, the muscle cells, they contain proteins such as actin and myosin. Also you have blood plasma also containing specific proteins such as albumin. Okay, those are used for transferring certain things in the blood. So in the blood plasma you expect to find these proteins. So they are the functions of proteins here: build up, bodybuilding. Okay, they are for bodybuilding. So they maintain the structure of the body. Also they're involved in tissue repair. Okay, damaged tissues. Okay, also they are used also for energy in the body, even though the body will prefer using carbohydrates for energy because carbohydrates are readily used. Okay, so many of the body proteins perform innumerable chemical reactions constantly taking place. Okay, very important. Proteins are very important because if you miss a certain protein in the body, it will result in a lot of disorders that maybe we don't even know how to treat them. Okay, so proteins are the molecular instruments in which genetic information is expressed. Okay, so quickly I explain this. So they are instruments through which genes express themselves. Okay, so you find that we have what we call DNA. Okay, and in this DNA you have genes. Okay, and these genes, they carry genetic information. Okay, so what will happen is that for that genetic information to be expressed, what does it mean to be expressed? Okay, when we say genetic information is being expressed, we're simply showing that it is showing its characteristic, the type of trait in that genetic information. So this genetic information is found in the DNA. Okay, and this is passed on from generation to generation, and we just inherit it. So what happens is this: for this genetic information to be used or to be expressed, the body uses a certain substance that we call mRNA. This is messenger RNA. So it will copy the information. So the information on DNA is copied to this messenger RNA. and this will now leave the nucleus. Okay, to leave the nucleus, it finds a certain molecule called the ribosomes. Okay, it attaches itself to the ribosomes. Okay, and then what happens is that these ribosomes now, ribosomes are for what? Protein synthesis. Okay, ribosomes are for protein synthesis, making of proteins. So what will happen? The ribosomes now, how does it make these proteins? We just know a ribosome, they are for protein synthesis, but then how do they make the proteins? Okay, they follow the information coming from messenger RNA, which came from DNA. Okay, so this information comes from here, and then it will give the instruction what type of proteins to be formed, and as a result here, it knows what type of amino acid to add to this amino acid to add to this amino acid, and later on you get what? A protein. Okay, so that's why we're saying that proteins are simply instruments in which genetic information is expressed. Okay, so I do hope that makes sense. Okay, so classification of proteins are classified in many ways. Okay, we can classify them in many ways, and we have what we call simple, and we also have conjugated. Simply here, simple meaning these only contain amino acids, nothing else. It's just a chain of many amino acids, nothing else. But then we have what we call conjugated. So conjugated, it simply means it's not just amino acids, it's not just a protein alone, but then something else will be added to it. Maybe another biomolecule is being added to it. For example, um, other biomolecules that can be added are lipids. Okay, you can add a lipid to a protein. What do you get? Lipoprotein. Lipo for lipid, and then protein. You can add a nucleic acid. Okay, you can add a nucleic acid. What do you get? Nucleoproteins. When you add glucose, you get glycoproteins. So these are called conjugated. Okay, conjugated proteins. Okay, so example. So composition. So based on the composition means what makes up that protein. Okay, if the only thing that is making up that protein are amino acids, it's a simple protein. But then if that protein not only contains amino acids but other biomolecules such as lipids, nucleic acids, carbohydrates, these fall under the conjugated proteins. And here we have lipoproteins, nucleoproteins, and glycoproteins. Also, you can classify them based on the solubility. Okay, you can classify them based on the solubility. Simply means they are, how readily do they dissolve in water. Okay, how soluble are they in water. Okay, how soluble. So you have this protein called albumin. Okay, an example, you can get egg albumin or serum albumin, which is found in the serum. You can get this from the serum in the blood. Okay, so this type of amino acid, protein, is soluble in water. So you can classify them. There are those that will be soluble and those that are not readily soluble in water. And the good part about albumin also, it coagulates by heating. So I find that when you boil an egg, find that it coagulates. Okay, boiled eggs, because that white thing here, it's albumin. Okay, albumin is a protein. What type of protein? A soluble protein. Okay, so you have others that are insoluble. Okay, you have other proteins that are basically insoluble. So you have globulins. Okay, these are also present in the serum. You can also find them in the muscle and even other tissues. These are not soluble in water, sparingly soluble in water. Okay, they do not dissolve readily in water. Okay, but they can dissolve in dilute salt solution. Okay, so according to solubility, others dissolve readily in water, others do not, but they can dissolve into a dilute soluble, salt solution. Okay, those are globulins. Okay, then you also have now another category of proteins called histone. So a histone, okay, so these um, histones, it's just it's a histone is a nucleoprotein. What we call a nucleoprotein, what we mean is that it is actually made up of a nucleic acid and proteins. Okay. So in a protein, so you find that you have these proteins, okay, called histones. Then what happens? The DNA just wraps around it, okay, making form something like beads. So you can find them in tissues such as the thymus, the pancreas, and these also fall under the soluble protein because they're soluble in water. So we're still looking at solubility, okay. Okay, so we move further. Also, you can classify them. This is the second classification, okay. The second classification based on the overall shape, okay, the overall shape. So you have the fibrous proteins. The molecule is constituted by several coiled cross-linked polypeptide chains, okay. Those that are fibrous, okay, they are fibrous, okay. And the examples, you have the collagen proteins. These are fibrous in nature. You also have the elastins. You also have the keratins, okay. So collagen is a major protein that you find in connective tissue. Elastins, elastin, they when you hear elastin, you think of elastic tissues, okay. So you find these elastins in tendons, arteries, or any other elastic tissue. Keratins, protein that makes up the hair and the nails. So all these fall under what? Fibrous protein. So you also have globular proteins. Globular simply means these are ovoid in shape, okay, not as the fibrous ones. Fibrous ones, they some that is fibrous, that's cross-linked, coiled, something like that. But globular proteins are simply ovoid in shape, and also they are soluble in water. They make up most of the enzymes, okay, and also oxygen carrying proteins and hormones. So oxygen carrying proteins, here we're talking about hemoglobin and most hormones, okay. Also, under globular proteins, you have these other proteins like albumin, globulins, and histone. They also fall under, because based on their shape, they are okay, globular, okay, based on the shape. Also, based on their what we call biological function. So there are those that have got the function of acting as enzymes, okay. You have proteins that have the function of acting as enzymes. Okay, so most enzymes are simply what? Protein. An enzyme is just a biological catalyst. Others are involved in regulatory uh, function. They regulate, okay, and these you have hormones. So regulation is just controlling, okay. The hormones, you can talk of here, insulin. What does it regulate? Blood sugar, okay, with its counterpart, glucagon, okay. Others, they simply bind to the DNA to regulate the process of either transcription, okay. So also other proteins are involved in transportation, okay, transporting substances, okay. For example, hemoglobin transports oxygen in the red blood cells. Myoglobin transports oxygen, but then, so these have the same function, but then hemoglobin, you find it in the red blood cells, okay, but then myoglobin is in the muscle. You also have another protein that is involved in transportation of iron. This is called transferrin, okay. Then you have another protein now that not only it doesn't transport the iron, but then it stores it in the liver, and this is called ferritin. So here it says there some are involved in transportation, some are involved in storage. Some okay, so coordinated motion. Proteins are a major component of muscle, okay. So find that in the muscle there, okay, there are two kinds of protein filaments. So the proteins that are found in the muscles usually are called filaments, okay, and they are for what? Muscle contraction, okay. A coordinated motion where one muscle has to contract where the other one has to relax. So this is some form of coordination, okay. So you can have maybe this joint which is here, the joint which is at the elbow here, the elbow joint. So there will be a protein here, there will be another protein here. These are muscles, right? So according to the types of protein here, they have to provide this coordination, okay, that you can flex your arm properly. So under coordinated motion, you have certain proteins, okay, and you can think of actin, myosin, okay, many types, okay. We'll see them as we go further. Some offer mechanical support, okay, because of their nature having high tensile strength, okay, so they offer support, okay. The examples are collagen and many fibrous protein. They do offer this mechanical support. Some in immune protection, okay. Most of the most of the proteins, okay, they are for what? Immune protection. Not most, but we know about what we call antibodies. We have this type of proteins, immunoglobulins, okay, immunoglobulins that are specific for immune. They're called immunoglobulins. These are proteins, but then they are for immune protection. Okay, so antibodies, highly specific proteins that distinguish between self and non-self. So they're able to recognize these what we call antigens. They will recognize something that belongs to the body and something that does not. Okay, and then they provide protection. So they will recognize it. Okay, so they can distinguish between self and non-self substances in the body. Some are involved in generation and transmission of nerve impulses. Okay, you have certain proteins that are involved um, in the response of nerve cells to specific stimuli. Okay, and these proteins, meaning how do they tell the stimuli? Because they've got because of the receptors, and some of these receptors are just made up of proteins. Okay, control of growth and differentiation. Okay, controlled sequential expression of genetic information. We've already talked about that. How genetic expression is. Okay, so now we go to something very important. That is the organization. Okay, the organization of these proteins. So the way the protein is organized. Okay, so when we look at organization, even in a company, you find that you have got maybe, just to give an example, organization, we're looking at how something is built, starting from the primary. Okay, maybe let's talk about it in terms of education, education systems. Okay, so you have as you're starting educational system, you have maybe you go to a kindergarten. Okay, maybe you start with baby classes and so on and so forth. After those baby classes, okay, just call them baby classes. After that, they graduate, they go into primary, which starts from grade 1. Then after that, they go into junior secondary, which is grade 8. Junior secondary, they are now senior secondary. Senior secondary, now you finish and they go into tertiary. Okay, tertiary, go into quaternary. Okay, so this is how these proteins also begin. Okay, so we'll look at this. So the first, or maybe level 1, in the way these proteins are made, okay, it's called, so the first structure that is made during protein synthesis, it is called a primary structure. Now, what is a primary structure? It is a linear sequence of amino acid residues. Okay, so we notice here what I was explaining, that each amino acid is added to the next amino acid by what? The ribosomes. Okay, like this. So a linear sequence meaning a straight line. So when the ribosomes, okay, produce this straight line chain of amino acids, it is called the primary structure. Okay, and for it to qualify as a primary structure, it needs to contain at least between 50 and 2,000 amino acid residues for it to qualify as a primary structure. Okay, and so the amino acid composition of a peptide chain has a profound effect on the physical and chemical. So here what is being said is that once this chain is formed, okay, it is what will determine the physical as well as the chemical properties of the protein. So what determines here? This primary structure, it is primary and the first level, but it is the most important level here, the primary structure, because it is what will give the physical and chemical properties of the protein once it is formed. Okay, because any mistake that will happen here, mostly these are irreversible. Okay, and maybe these, most of them are inherited mutations. Okay, protein rich in polyamino acids. So for example, amino acids, if this chain of amino acids, if the chain of amino acids contains a lot of what polar amino acids, meaning the whole protein will become polar. So depending on the properties of those amino acids, they will determine the physical and chemical properties of the bigger, large substance, the protein itself. Proteins that are rich in aliphatic or aromatic amino groups, they're usually insoluble. So it all comes from the type of amino acids inside the protein. Okay, it's what creates these things. So apart from this, now after the primary structure is formed, they graduate now to secondary structures. This is called a local structure. Okay, polypeptide chain, which is determined mainly by hydrogen bonds. Okay, what are hydrogen bonds? These are interactions that occur between the carbonyl oxygen group of one peptide bond and the amide. Okay, and we'll see how these. So among the secondary structures, you have two common ones. You have two types here. You have the alpha helix. The alpha helix is a rod-like structure which with peptide chains tightly coiled. Okay, like this, something like this. Okay, okay, and then you have these hydrogen bonds that are formed, maybe there, maybe there. Okay, like that. Okay, so with peptide chains tightly coiled and the side chains of amino acids residues extending outward the axis of the spiral. Okay, let's look at a better. So each amino acid differs from the next amino acid. So the hydrogen bond will occur. So each amino carbonyl group is hydrogen bonded to amino acid that is 4 residues away. Okay, so 4 residues away, it means this, there should be at least 4 amino acids. Okay, so 4 residues away, meaning these now can form. Okay, so 4 residues away from the same chain. Okay, so you have also the beta pleated sheets, hydrogen bonds formed between peptide bonds in different chains. So you have a parallel and antiparallel chain. So here we have the diagrams. So according to the protein structure, you have so after the primary amino acid sequence is formed, which is the primary structure, the protein can also assume a secondary structure, and there are two types here. You have the alpha helix, which is here. Okay, you have the alpha helix, and you also have the beta pleated sheets here. Okay, so this is how the alpha helix actually looks like. Okay, then you have the beta pleated sheets consisting of parallel and antiparallel chains. Okay, so you have maybe a chain that is like this, and you have maybe another chain that is like this, parallel and antiparallel, and then these can form bonds like that. Then the alpha helix is one chain that is coiled like this, but then where does the hydrogen bonding form? The hydrogen bonds can just form right here. So you can find that you have a hydrogen bond formed here, there, 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 there. Okay, and it makes that type of a thing. Okay, this is a hydrogen bond. Okay, so now what we are supposed, so this is a quaternary structure. Okay, so like example, this is an alpha helix, so it coils itself and you have bonds forming like that. So tertiary structure, now this is a three-dimensional folded biologically active. So after the secondary structure, the structure that will come up is a tertiary structure. Now at this point here, the protein it's biologically active. It is biologically active. It can perform its intended function, okay, at the tertiary structure level. It will be able to perform the intended function. So at primary, secondary, it's not yet biologically active until it reaches the tertiary structure. So the structure reflects the overall shape of the molecule. The structure here stabilized by many interactions, okay, between the side. So these interactions that you are expected to see in the tertiary structure, questions come here. It has covalent disulfide bonds that are formed between amino acids that contain a sulfur, so it's called disulfide. You also have hydrogen bonds that are also found in the secondary structure. You also have salt bridges now, okay, that are formed between amino acid that has an acid group and an amide group. Once these salt and a base, I mean an acid and a base, they make up a salt. That's why they are called salt bridges, because this COO is acid, amide is base, okay. Right, other interactions occur between hydrophobic what amino acids, and these are called hydrophobic interactions. For example, side chains of amino acids like leucine, valine, and phenylalanine. Also, you have chains that side chains of tryptophan, arginine. These also serve as hydrogen donors, okay. So for those that will be involved in the salt bridges, you have lysine. So remember, salt bridges are formed between amino acid that has an acidic part, acidic side chain, and one that has an amide. Okay, for example, aspartic acid has an acidic part, also glutamic acid has an acidic part. But tyrosine, histidine, they have an amide part. So once these they interact, you make up what we call salt bridge. Okay. Two oppositely charged amino acids such as glutamate, this is what is being explained here. Okay, I've already explained that. Quaternary structure now. Quaternary structure is very complex compared to the other structures. After the primary structure is number one, secondary, tertiary, now the quaternary structure. Okay, it's very complex, and this one consists of maybe two or more tertiary what structures, okay, complex, the assembly of two or more separate peptide chains, okay, non-covalent or covalent interactions. So for example, there are two types of the quaternary structure. You have the homogeneous one. So if those tertiary structures are the same, it's called homogeneous, meaning they have identical subunits. For example, which example can I give? Okay, let's look at heterogeneous, one which has different subunits of tertiary structures. Is what we call subunit A and subunit B. Same thing, hemoglobin, it has four chains. Two of them are alpha subunit, two of them are beta. So this is for under heterogeneous. Protein purification. Protein can be purified based on charge size, binding properties, and solubility. Okay. Then protein modification. This is where a protein is modified after it's been synthesized, and this occurs when through addition of either an acetyl group, either an hydroxyl, okay. For example, when acetyl group is added, the process called acetylation, and such proteins, after they are modified in that way, you find that they become more resistant to degradation. Sometimes you can add the hydroxyl group we come up with hydroxylation process here, where proline can be you can add this hydroxyl group to proline, and here what the body comes up is called hydroxyproline, which stabilizes collagen fiber. So once the proteins are modified, they will have a certain function that is very important for the body. Okay, you also have, for example, if let's look at this disorder scurvy. This comes about due to insufficient hydroxylation of collagen. Okay, once collagen, that hydroxylation does not take place, the person will end up having scurvy. Okay, and this hydroxylation becomes insufficient simply because of lacking vitamin C. Also, in vitamin K deficiency, you find that carboxylation is not taking place, and glutamate here is what is involved in this carboxylation process. So find that it's not, if carboxylation doesn't take place, carboxylation of glutamate in prothrombin, the body won't be able to form properly clotting factors, and there will be hemorrhage, right? Which is excessive bleeding because of lacking certain protein factors due to deficiency of vitamin K. So as a result, deficiency of vitamin K, meaning there will be insufficient hydroxylation. Deficiency of vitamin C, there will be insufficient hydroxylation. So you see how these post-translational modifications are important as well. When you add the sugar, you add the protein will become hydrophilic, meaning water loving, it can dissolve in water. But when you add a fatty acid, it becomes hydrophobic. Okay, so you have digestive enzymes. Okay, these are always produced in their inactive form, then they are then inactivated by some process. You also have blood clotting proteins soluble, such as fibrinogen. For them to operate, they have to be converted to something insoluble for them to form a clot. All these so modification are just finishing touches. The protein has already been formed. Okay, it's biologically active, just a finishing touch to add on. Okay, so account of much versatility, precision, and elegance of the protein. Okay, now the role of chaperones, they simply mark those proteins that haven't been formed properly. They mark them down either for destroying them or remodification. Okay, that's what the chaperones do. Denaturation of protein is when the protein loses its function. Okay, it's denatured. Okay, and this can occur when that protein is brought in certain environments such as oxidation, proteolysis. Okay, all these denaturation. So according to the type of environment, the protein, sometimes even just high temperature will denature a protein. Okay, so denaturation, finite lifetime. Okay, so it's not, sometimes it is reversible, sometimes it is not reversible. So this denaturation, so the structure, this is when the protein loses its structure and it won't perform its function. The shape of anything determines its function. So denaturation, the destruction of the high level structure starting from the primary, tertiary, and quaternary. So here there's the primary is not included, because once a protein is denatured, it can lose all these structures from from secondary, tertiary, and quaternary, but the primary structure is always maintained. Okay, it was maintained, so that when that denaturing substance is removed, it can now go back to form the secondary, the tertiary, as well as the quaternary structure. Okay, so protein, once it's denatured, it will lose its native physiochemical and biological properties. Okay, since the bonds that stabilize it are broken, okay, it will unfold itself and it will remain in that unfolded state, but the primary structure is maintained. Okay, the denatured protein may retain its biological activity by simply refolding itself when the denaturing agent is removed. So here now it just talks about the clinical application. Okay, and its clinical application looks at how these proteins are used clinically. What's the importance of learning about these proteins? So utilization of amounts of proteins found in urine. Okay, these are samples that come to the lab: serum, cerebrospinal fluid. Okay, these we assess certain proteins in them. Okay, so there are certain proteins that we expect in other substance in certain fluids, and we don't expect them. So appearance of proteins like albumin and globin in urine simply means there's uh, some damage to the kidney. Okay, there's been an impairment because proteins are not supposed to be found in the urine. Okay, so we can use that to diagnose the kidney functions. Okay, so in kidney functions we can check for certain proteins that are found in shouldn't be found in urine, because these are, in urine these proteins are large. Okay, then how do they pass through the glomerulus? It means it's damaged somehow. Okay, abnormal proteins also found in plasma. Okay, meaning they're filtered. You have Benz-Jones condition, protein in urine, most associated with multiple myeloma. Okay, so recognition of such protein in urine may be used for diagnosis of certain diseases. Also, CSF, your turbidity, once it increases turbidity increases in the CSF, shows that there's an increase in globulins. An increase in gamma globulins is observed in the case of multiple sclerosis and neurosyphilis. Okay, so that's on the clinical application of proteins. Okay, so here it says vertebrates are aerobic organisms with closed circulatory system. Okay, so mechanism of extracting O2 from the air or water releases carbon dioxide. So oxygen for aerobic respiration, release of energy. Okay, let's see if we can. Okay, so heme proteins, there are two of them, myoglobin and hemoglobin. These are for oxygen transportation. So let's look at what happens in sickle cell disease. This is an inherited disorder where a person inherits this distorted red blood cells, and these will mean that there will be limited blood flow. Okay, because these distorted red blood cells, they will occlude certain blood vessels. The blood won't be able to flow. Okay, so just some maybe clinical presentation. Okay, intermittent episodes of hemolytic anemia. I mean, the red blood cells, since they have a sickle shape, so they end up having a sickle shape like this. Okay, it's easy for one to stack on the next one like that, and this happens in the blood vessel. Blood won't be able to flow. Okay, and the same red blood cells also, they are prone to damage. So it causes hemolytic anemia where there's excessive destruction of these red blood cells. This will cause impaired growth, increased susceptibility to infections, and multiple organ damage. Sickle cell disease. Okay. Okay, so the cause of this sickle cell disease is a single point mutation. Okay, a single point mutation where glutamate, okay, replaces valine. Okay, so it's just here. It's where the problem is. Okay, here. So where the problem is. In this chain of amino acids, there is a swap. Instead of glutamate, then valine appears. Okay, this is what happens in sickle cell disease. Okay, the mutation occurs just 1 point, so it's called a single point. Only 1 point here. Okay, so valine fitting in a pocket that is exposed upon release of oxygen. So you find that because of this, when the bodies or maybe the blood is deoxygenated, it is less soluble. Okay, long filamentous polymers precipitate. Okay, and this causes the morphology of the red blood cell to be sickle shaped. So there is a homozygous inheritance and also the heterozygous. Heterozygous, these are just carriers. They don't show any symptom, but then once you inherit homozygous nature, you become sickle shaped. Okay. Right, that's on sickle cell. We could do the other ones next time.